Anatomy of flowering plants. You can very easily see the structural similarities and variations in the external morphology of the larger living organisms, both plants and animals. Similarly, if we were to study the internal structure, one also finds several similarities as well as differences. This chapter introduces you to the internal structure and functional organization of higher plants. Study of internal structures of plant is called anatomy. Plants have cell as the basic unit. Cells are organized into tissues and in turn the tissues are organized into organs. Different organs in a plant show differences in their internal structure. Within angiosperms, the monocots and dicots are also seen to anatomically different. Internal structures also show adaptations to diverse environments. 6.1 The tissues. A tissue is a group of cells having a common origin and usually performing a common function. A plant is made up of different kinds of tissues. Tissues are classified into two main groups, namely meristematic and permanent tissues, based on whether the cells being formed are capable of dividing or not. 6.1.1 Meristematic tissue. Growth in plants is largely restricted to specialized regions of active cell division called meristems. Plants have different kinds of meristems. The meristems which occur at tip of roots and shoots and produce primary tissues are called apical meristems. Root apical meristems occupies the tip of a root, while the shoot apical meristem occupies the distance most region of the stem axis. During the formation of leaves and elongation of stem, some cells left behind from shoot apical meristem constitute the axillary bud. Such buds are present in the axils of leaves and are capable of forming a branch or a flower. The meristem which occurs between mature tissue is known as intercalary meristem. They occur in grasses and regenerate parts removed by the grazing herbivores. Both apical meristems and intercalary meristems are primary meristems because they appear early in life of a plant and contribute to the formation of primary plant body. The meristem that occurs in the mature region of fruits and shoots of many plants, particularly those that provides woody axis and appear later than primary meristem, is called the secondary or lateral meristem. Their cylindrical meristems, vascular cambium, interfascicular cambium, and core cambium are examples of lateral meristems. These are responsible for producing the secondary tissues. Following divisions of cell in both primary as well as secondary meristems, the newly formed cells become structurally and functionally specialized and lose the ability to divide. Such cells are termed permanent or mature cells and constitute the permanent tissues. During the formation of the primary plant body, specific regions of the apical meristem produce dermal tissues, ground tissues, and vascular tissues. 6.1.2 Permanent tissues. The cells of the permanent tissues do not generally divide further. Permanent tissues, having all cells similar in structure and function, are simple tissues. Permanent tissues having many different types of cells are called complex tissues. Simple tissues. A simple tissue is made up of only one type of cells. The various simple tissues in plants are parenchyma, collenchyma, and sclerenchyma. Parenchyma forms the major components within organs. The cells of the parenchyma are generally isodiametric. They may be spherical, oval, round, polygonal, or elongated in shape. The walls are thin and made up of cellulose. They may either be closed, packed, or have small intercellular spaces. The parenchyma performs various functions like photosynthesis, storage, secretion. The collenchyma occur in layers below the epidermis in most of the dichotomous plants. It is found either on a homogeneous layer or in patches. It consists of cells which are much thickened at the corners due to deposition of cellulose, hemicellulose, and pectin. Collenchymatous cells may be oval, spherical, or polygonal and often contain chloroplast. These cells assimilate food when they contain chloroplast. Intercellular spaces are absent. They provide mechanical support to the growing parts of the plant, such as young stem and petiole of leaf. Sclerenchyma consists of long, narrow cells with thick and lignified cell wall having few or numerous pits. They are usually dead and without protoplast. On the basis of variation in form, structure, origin, and development, sclerenchyma may either fibers, or sclerites. The fibers are thick walled, elongated, and pointed cells, generally occupying in groups in various parts of the plant. The sclerites are spherical, oval, or cylindrical, highly thickened dead cells with very narrow cavities lumen. These are commonly found in the fruit walls of nuts, pulp of fruits like guava, pear, and sapota, seed coats of legumes, and leaves of tea. Sclerenchyma provides mechanical support to organs. Complex tissues 6.1.2.2. The complex tissues are made up of more than one type of cells, and these work together as a unit. Xylem and phloem constitute the complex tissues in plants. Xylem functions are the conducting tissues for water and minerals from root to the leaves and stems. It also provides mechanical strength to the plant parts. It is composed of four different kinds of elements, namely tracheids, vessels, xylem, fibers, and xylem parenchyma. Gymnosperms lack vessels in their xylem. Tracheids are elongated or tube-like cells with thick and lignified walls and tapering ends. These are dead and are without protoplasm. The inner layers of the cell wall have thickenings which vary in form. In falling plants, tracheids and vessels are the main water transporting elements. Vessel is a long cylindrical tube like structure made up of many cells called members, each with lignified walls and a large central cavity. The vessel cells are also devoid of protoplasm. Vessel members are interconnected through perforations in their common walls. The presence of vessels is a characteristic feature of angiosperms. Xylem fibers have thickened walls and obliterated central lumens. These may either be septate or aseptate. Xylem parenchyma cells are living in thin walled and their cell walls are made up of cellulose. They store food materials in the form of starch or fat and other substances like tannins. The radial conduction of water takes place by ray parenchymatous cells. Primary xylem is of two types, protoxylem and metaxylem. The first formed primary xylem elements are called protoxylem and the later formed are called metaxylem. In stems, the protoxylem lies towards the center pith and the metaxylem lies towards the periphery of the organ. This type of primary xylem is called endark. In roots, the protoxylem lies towards periphery and metaxylem lies towards the center. Such arrangement of primary xylem is called exarch. Phloem transports food materials, usually from leaves to the other parts of the plant. Phloem in angiosperms is composed of seed tube elements, companion cells, phloem parenchyma, and phloem fibers. Gymnosperms have albuminous cells and seed cells. They lack sieve tubes and companion cells. Sieve tube cells elements are also long, tube-like structures arranged longitudinally and are associated with the companion cells. Their end walls are perforated in a sieve-like manner to form the sieve plates. A mature sieve element possesses a peripheral cytoplasm and a large vacuole but lacks a nucleus. The functions of the sieve tubes are controlled by nucleus of companion cells. The companion cells are specialized parenchymatous cells which are closely associated with sieve tube elements. The sieve tube elements and companion cells are connected by pit fields present between the common longitudinal walls. The companion cells help in maintaining the pressure gradient in the sieve tubes. Phloem parenchyma is made up of elongated tapering cylindrical cells which have dense cytoplasm and nucleus. The cell wall is composed of cellulose and has pits through which plasmodesmatal connections exist between the cells. The phloem parenchyma stores food material and other substances like resins, latex, and mucilage. Phloem parenchyma is absent in most of the monocotyledons. Phloem fibers, bast fibers, are made up of sclerenchymatous cells. These are generally absent in primary phloem but are found in secondary phloem. These are much elongated, unbranched, and half pointed needle like apex. The cell wall of phloem fibers is quite thick. At maturity, these fibers lose their protoplasm and become dead. Phloem fibers of jute, flax, hemp are used commercially. The first form primary phloem consists of narrow sieve tubes and is referred to as protophlloem, and the later formed phloem have bigger sieve tube and is referred to as metaphloem. 6.2 The tissue system. We are discussing types of tissues based on the types of cell present. Let us now consider how tissue vary depending on the location in the plant body. The structure and function would also be dependent on the location. On the basis of the structure and location, there are three types of tissue systems. These are the epidermal tissue system, ground or fundamental tissue system, and the vascular or conducting tissue system. 6.2.1 Epidermal tissue system. The epidermal tissue system forms the outermost covering of the whole plant body and comprises epidermal cells, stomata, and the epidermal appendages, the trichomes and hairs. The epidermis is the outermost layer of primary plant body. It is made up of elongated, compactly arranged cells which forms a continuous layers. Epidermis is usually single layer. Epidermal cells are parenchymatous with a small amount of cytoplasm lining the cell wall and a large vacuole. The outside of the epidermis is often covered with a waxy thick layer called cuticle, which prevents the loss of water. Cuticle is absent in roots. Stomata are structure present in the epidermis of leaf. Stomata regulate the process of transpiration and gaseous exchange. Each stoma is composed of two bean-shaped cells known as guard cells, which enclose stomatal pore. In grasses, the guard cells are dumbbell shaped. The outer walls of guard cells away from the stomatal pore are thin and inner walls towards the stomatal pore are highly thickened. The guard cell possesses chloroplast and regulate the opening and closing of stomata. Sometimes a few epidermal cells in the vicinity of the guard cell become specialized in this shape and size and are known as subsidiary cells. The stomatal aperture, guard cells, and the surrounding subsidiary cells are together called stomatal apparatus. The cells of epidermis bear a number of hairs. The root hairs are unicellular elongations of an epidermal cell and help absorb water and minerals from the soil. On the stem, the epidermal hairs are called trichomes. The trichomes in the shoot system are usually multicellular. They may be branched or unbranched and soft or stiff. They may even be secretory. The trichomes help in preventing water loss due to transpiration. 6.2.2 The ground tissue system. All tissues except epidermis and vascular bundles constitute the ground tissue. It consists of a simple tissue such as parenchyma, collenchyma, and sclerenchyma. Parenchymatous cells are usually present in the cortex, periacycle, pith, and medullary rays, in the primary stems and roots. In the leaves, the ground tissue consists of thin-walled chloroplast containing cell and is called mesophyll. 6.2.3 The vascular tissue system. The vascular system consists of complex tissues, the phloem and the xylem. The xylem and the phloem together constitute vascular bundles. In dicotyledonous stems, cambium is present between phloem and xylem. Such vascular bundles, because of the presence of cambium, possesses the ability to form secondary xylem and phloem tissues and hence are called open vascular bundles. In the monocotyledons, the vascular bundles have no cambium present in them. Hence, since they do not form secondary tissue, they are referred to as closed. When xylem and phloem within a vascular bundle are arranged in alternate manner along the different radii, the arrangement is called radial, such as in roots. In conjoint type of vascular bundle, the xylem and phloem are jointly situated along the same radius of vascular bundles. Such vascular bundles are commonly in stems and leaf. The conjoint vascular bundle usually have the phloem located on the outer side of xylem. 6.3 Anatomy of dicotyledonous and monocotyledonous plant. For a better understanding of tissues, organization of root, stem, and leaves, it is convenient to study the transverse section of mature zone of these organs. 6.3.1 Dicotyledonous root. Look at the figure 6.6a. It shows the transverse section of the sunflower root. The internal structure organization is as follows. The outermost layer is epiblema. Many of the cells of epiblema protrude in form of unicellular root hairs. The cortex consists of the several layers of thin-walled parenchyma cells with intercellular spaces. The innermost layer of the cortex is called endodermis. It comprises a single layer of barrel-shaped cells without any intercellular spaces. The tangential as well as radial walls of the endodermal cells have a deposition of water-impermeable waxy material suberin in the form of Casparian strips. Next to endodermis lies a few layers of thick-walled parenchymatous cell referred to as pericycle. Initiation of lateral roots and vascular cambium during the secondary growth takes place in these cells. The pith is small or inconspicuous. The parenchymatous cells, which lie between the xylem and the phloem, are called conjunctive tissues. These are usually 2 to 4 xylem and phloem patches. Later, a cambial ring develops between the xylem and phloem. All tissues on the inner side of the endodermis, such as pericycle, vascular bundles, and pith, constitute the stele. 6.3.2 Monocotyledonous root. The anatomy of monocot root is similar to the dicot root in many respects. It has epidermis, cortex, endodermis, pericycle, vascular bundles, and a pith. As compared to the dicot root, which has fewer xylem bundles, there are usually more than 6 polyarch xylem bundles in the monocot root. Pith is large and well developed. Monocotyledonous roots do not undergo any secondary growth. 6.3.3 Dicotyledonous stem. The transverse section of a typical young dicotyledonous stem shows that the epidermis is the outermost protective layer of the stem, covered with a thin layer of cuticle. It may bear trichomes and few stomata. The cells arranged in multiple layers between epidermis and pericycle constitute the cortex. It consists of three subzones: the outer hypodermis, consisting of a few layers of collenchymatous cell just below the epidermis, which provide mechanical strength to the young stem. Cortical layers below hypodermis consist of rounded, thin-walled parenchymatous cell with conspicuous intercellular spaces. The innermost layer of the cortex is called endodermis. The cells of the endodermis are rich in starch grains, and the layer is also referred to as starch sheath. Pericycle is present on the inner side of endodermis and above the phloem in the form of semilunar patches of sclerenchyma. In between the vascular bundles, there are few layers of radially placed parenchymatous cells, which constitute medullary rays. A large number of vascular bundles are arranged in a ring. The ring arrangement of vascular bundles is characteristic of dicot stem. Each vascular bundle is conjoint, open, and with in dark protoxylem. A large number of rounded parenchymatous cells with large intercellular spaces which occupy the central portion of the stem constitute the pith. 6.3.4 Monocotyledonous stem. The monocot stem has sclerenchymatous hypodermis, a large number of scattered vascular bundles, each surrounded by sclerenchymatous bundle sheath and a large conspicuous parenchymatous ground tissue. Vascular bundles are conjoint and closed. Peripheral vascular bundles are generally smaller than the centrally located ones. The phloem parenchyma is absent and water-containing cavities are present within the vascular bundles. Dorsiventricle leaves. 6.3.5 The vertical section of a dorsiventral leaf through the lamina show three main parts, namely epidermis, mesophyll, and vascular system. The epidermis, which covers both the upper surface adaxial epidermis and lower surface abaxial epidermis of the leaf, has conspicuous cuticle. The abaxial epidermis generally bears more stomata than the adaxial epidermis. The latter may even lack stomata. The tissue between the upper and the lower epidermis is called mesophyll. Mesophyll, which possesses chloroplast and carry out photosynthesis, is made up of parenchyma. It has two types of cells, the palisade parenchyma and the spongy parenchyma. The adaxially placed palisade parenchyma is made up of elongated cells, which are arranged vertically and parallel to each other. The oval or round and loosely arranged spongy parenchyma is situated below the palisade cells and extends to lower epidermis. There are numerous large spaces and air cavities between these cells. Vascular system includes vascular bundles, which can be seen in the veins and midrib. The size of vascular bundles are dependent on the size of the veins. The veins vary in thickness in the reticulate venation of the dicot leaves. The vascular bundles are surrounded by a layer of thick-walled bundle sheath cells. Look at figure 6.8a and find the position of xylem in the vascular bundle. Isobilateral monocotyledonous leaf. The anatomy of isobilateral leaf is similar to that of dorsiventral leaf in many ways. It shows that the following characteristic differences in an isobilateral leaf: the stomata are present on both the surfaces of epidermis, and the mesophyll is not differentiated into palisade and spongy parenchyma. In grasses, certain adaxial epidermal cells along the veins modify themselves into large empty colorless cells. These are called bulliform cells. When the bulliform cells in the leaf have absorbed water and are turgid, the leaf surface is exposed. When they are flaccid due to water stretch, they make the leaves curl inwards to minimize water loss. The parallel venation in monocot leaf is reflected in near similar sizes on vascular bundle except in main veins as seen in vertical sections of the leaves. 6.4 Secondary growth. The growth of the roots and the stem in the length with the help of apical meristem is called primary growth. Apart from the primary growth, most dicotyledonous plants exhibit an increase in girth. This increase is called the secondary growth. The tissues involved in secondary growth are the two lateral meristems, the vascular and the core cambium. The vascular cambium 6.4.1. The meristematic clear that is responsible for cutting off vascular tissues, xylem and phloem, is called vascular cambium. In the young stem, it is present in patches as a single layer between xylem and phloem. Later, it forms a complete ring. 6.4.1.1 Formation of cambial ring. In dicot stem, the cells of cambium present between primary xylem and primary phloem is the intrafascicular cambium. The cells of medullary rays adjoining this interfascicular cambium become meristematic and form the interfascicular cambium. Thus, a continuous ring of cambium is formed. 6.4.1.2 Activity of cambial ring. The cambial ring becomes active and begins to cut off new cells both towards the inner and the outer sides. The cell cut off towards pith mature into secondary xylem and the cells cut off towards periphery mature into secondary phloem. The cambium is generally more active on the inner side than on the outer side. As a result, the amount of secondary xylem produced is more than secondary phloem and soon forms a compact mass. The primary and secondary phloem gets gradually crushed due to continued formation and accumulation of secondary xylem. The primary xylem, however, remains more or less intact in or around the center. At some places, the cambium forms a narrow band of parenchyma, which passes through the secondary xylem and the secondary phloem in radial directions. These are the secondary medullary rays. 6.4.1.3 Springwood and autumnwood. The activity of cambium is under the control of many physiological and environmental factors. In temperate regions, the climatic conditions are not uniform throughout the year. In the spring season, cambium is very active and produces a large number of ciliary elements having vessels with wider cavities. The wood formed during this session is called spring wood or early wood. In winter, the cambium is less active and forms few xylary elements that have narrow vessels, and this wood is called autumn wood or late wood. The spring wood is lighter in color and has lower density, whereas the autumn wood is darker and has higher density. The two kinds of woods that appear as alternate concentric ring constitute an annual ring. Annual ring seen in a cut stem give an estimate of age of a tree. 6.4.1.4 Hardwood and sapwood. In older trees, the greater part of the secondary xylem is dark brown due to deposition of organic compounds like tannins, resins, oil gums, aromatic substances, and essential oils in the central or innermost layer of the stem. These substances make it hard, durable, and resistant to attack of microorganisms and insects. This region comprises dead elements with highly lignified wall and is called hardwood. The hardwood does not conduct water, but it gives mechanical support to the stem. The peripheral region of the secondary xylem is lighter in color and is known as sapwood. It is involved in conduction of water and minerals from root to leaf. 6.4.2 Cork cambium. As the stem continues to increase in girth due to the activity of vascular cambium, the outer cortical and epidermis layer get broken and need to be replaced to provide new protective cell layers. And sooner or later, another meristematic tissue called corcambium or phellogen develops usually in cortex region. Phellogen is a couple of layers thick. It is made up of narrow, thin-walled and nearly rectangular cell. Phellogen cuts off cells on both sides. The outer cell differentiate into cork or phellum while the inner cell differentiate into secondary cortex or phelloderm. The cork is impervious to water due to suberin deposition in cell wall. The cells of secondary cortex are parenchymatous, phellogen, phellum, and phylloderm are collectively known as periderm. Due to activity of cork cambium, pressure builds upon remaining layers peripheral to phellogen, and ultimately these layers die and slow off. Bark is non-technical term that refers to all tissues exterior to vascular cambium, therefore including secondary phloem. Bark refers to number of tissue types with periderm and secondary phloem. Bark that is formed early in the season is called early or soft bark. Towards the end of the season, late or hard bark is formed. Name the various kind of cellular which constitute the bark. At certain region, the phyllogen cuts off closely arranged parenchymatous cells on the outer side instead of cork cell. These parenchymatous cells soon rupture the epidermis, forming a lens shape opening called lenticels. Lenticels permit the exchange of gases between the outer atmosphere and the internal tissues of the stem. These occur in most woody trees. 6.4.3 Secondary growth in root. In the dicot root, the vascular cambium is completely secondary in origin. It originates from the tissue located just below the phloem, bundles a portion of pericycle tissue above the protoxylem, forming a complete and continuous wavy ring. Further events are similar to those already described above for dicotyledon stem. Secondary growth also occurs in stem and roots of gymnosperms. However, secondary growth does not occur in monocotyledons.